Hello everybody, my name is Brandon Hong, and today I'm going to be talking to you guys how Trevor and I have found that when you express a 128 length polyglutamine repeat in Drosophila, the effects on the motor function is severe. It is on the order of magnitude of about 3 times. We have also found that a 16 length polyglutamine repeat shows little to no effect on Drosophila motor function. Now, previous studies have shown that when you increase the length of polyglutamine repeats, there is a change in Drosophila behavior. However, the link between the length of polyglutamine repeat you express and the degree of deterioration in the motor functions of the Drosophila has yet to be seen. So what we did was use human HTT DNA and put it into Drosophila. So what we did was we made a transgenic organism. And we varied the lengths of CAG repeats, which code for the glutamines, so that we could express HTT polyQ16, HTT polyQ128 in Drosophila. And those two groups here are our experimental groups. These two groups here are our control groups. So for our control, we used UAS polyQ, either 16 or 128. And we chose these control groups because we wanted to make sure that the UAS polyQ system, polyQ transgene, was not affecting Drosophila motor functions. So we actually did this this experiment in duplicate, where there's actually 2 separate sets of the same genotype. And we took the data and averaged them together to form the results here. The method that we use in our experiment is negative geotaxis. So what we did was we got a vial and we tapped the flies down to the bottom. The flies are in our vial and the flies are now to the bottom. And when we stop tapping, the flies go back up. And what we did was we counted 30 seconds using a video recorder, counted 30 seconds and counted the amount of flies that migrated 8 centimeters in 30 seconds. And we calculated a percentage for that replicate. And what we did was, as I said before, we have 2 replicates, so we averaged the percentages together to get these data shown here. As you can see, polyQ128 expressing shows a dramatic decrease in motor functions when compared to that of the control. Now, you may also notice that the error bar is also a little large, and that's because of the way we calculated error. So the way we calculated error was the distance from the average. Now, in one replicate for this experiment here in red of the expression 128 polyQ repeat in Drosophila, there was about 48% of the Drosophila able to migrate 8 centimeters in 30 seconds. Whereas in another replicate, 0 were able to migrate 8 centimeters in 30 seconds. So that means that the average percentage of flies able to migrate 8 centimeters in 30 seconds that were expressing the HTT polyQ, HTT 128 glutamine repeat was 24%. But that also means that the error was 24%. So if we had more time, we would have done more replicate testing on this group in particular, so that we were able to get a better average as well as a lower standard deviation. We also did the same for the 16 length polyglutamine expansion. And as you can see, there's little to no effects. In conclusion, you can see that long lengths of polyQ dramatically inhibit the motor functions of Drosophila on the order of about 3 times. And lower lengths of about 16 polyQs do not inhibit the motor functions of Drosophila at all. Now, for the future directions, we would of course like to create more replicate testing on the HTT, on the expressing Drosophila of HTT 128 polyQ. However, we would also like to see if the inhibition of motor functions increases gradually with increasing polyQ lengths, or if there's a threshold number where as soon as you hit the threshold number, you will see polyQ repeats you will see a dramatic decrease in motor functions. Of course, I would like to thank Professor Casey for her time, and Travis and Chiquita for their mentorship for this semester.